genomes collection. There's 495 genomes listed, and I've just tried to give a distribution, an approximate distribution of how these are according to their genome size. The average overall is about 50,000 base pairs. And you can see that there's quite a lot of small guys here, things that are around 5, 6, 7 kilobase pairs. There's a few relatively large ones here, 150-ish around there. The largest is one that's actually not in this collection, which is about 500 kb. But there's a predominance of genomes that are in this size range right here, around 50 kilobases, give or take. So between 35 and 70 kilobases is where the majority of these genomes lie. And these small guys here are probably something of an overrepresentation. In nature, these may not be as prevalent as these are in general. It's just that these guys are small and therefore much easier to sequence and therefore easier to, relatively speaking, to overpopulate the genome databases. And this group used to be much more prominent within this type of distribution than what we see at the moment. Now, incidentally, I should, if it's not already apparent, 500 or so is a number that is much, much smaller than 10 to the power of 31, right? In fact, this is almost certainly smaller than the number of bacterial genomes that have been sequenced, even though bacterial genomes are about 100 times the size. And so we have, I think, very much dragged our heels somewhat, let's say, in terms of characterizing and determining the genome sequences of bacteriophages, something that hopefully will change. There are some themes. I don't want to go through long lists and detailed maps of 495 genomes, but I do want to pull out some themes that I think are helpful in thinking about what functions these genomes encode and how they're organized. And so the first point I want to make, this is showing a genetic organization of a genome, which I believe is a phage called ES18, and is an enterobacteriophage. And the point that I want to make is that in this case, we know something about what many of the functions, or can predict what many of the functions of these genes do. And you see, first of all, in this part of the genome here that's annotated here as having heads and then tails, there's a long set of genes that are involved in making the proteins that compose the virion structures, heads and tails. And these genes are actually organized in an order, in a synteny, if you like, which is well conserved amongst many bacteriophages, even though the nucleotide and the amino acid sequences can be very, very varied, very different. But gene order in these genes that are involved in DNA packaging, the formation of heads, the capsids, the portals, the little doorways through which DNA is packaged and tails are attached, and the genes that are involved in the tail assembly, the so-called tape measure protein, the protein, the so-called major tail subunit, which forms the shaft of the tail as you see it, and various tail fibers here, tail fibers which almost certainly encode the specificity for the recognition of the host. They're encoded in this common gene order in this and many other phages of this type, and I'll explain that qualification in a minute. You can see that that set of genes occupies about half of this genome. And I told you the average size of these genomes, if we just throw out a number, was about 50,000 base pairs. The genes that are involved in the in the organization of these structural genes is quite commonly in the 20 to 25 kilobase span, just as a ballpark. There's some other gene functions that are noted down in this right-hand part of the genome here. I'll point out those that are involved in integration. So here is that integrase gene. It's one of those tyrosine integrases that I described earlier. There are genes that are involved in DNA replication. There are some genes that are involved in recombination, which is not uncommon in some of these bacteriophage genomes. This here is the regulatory function. It's the repressor gene for the transcriptional repressor, since this is a temperate bacteriophage. And most importantly, there are genes that specifically encode the functions for lysis of the host at that terminal stage of lytic growth. So we have structural genes in a well-conserved order, and we have the non-structural genes that are involved in replication, recombination, and regulation. But not every phage is like ES18, and not every phage adopts that organization. And this shows the map of another well-characterized E. coli phage called T4. It's much, much bigger. It's got a lot more genes. And the organization is a lot less tidy, if that's the right word. There is conservation of, there is the grouping of genes together on the genome that have common or related functions. Head genes here, some base plate tail functions here. But then you find assorted other structural proteins elsewhere in this genome as well. And so you can't simply look at well-conserved sets of genes in terms of their organization with regard to function. And so there's a number of different varieties. Just to expand that from those two genomes that I showed you, this is now looking at just maps, just rather gross maps of a set of mycobacteriophages, and the top, however many of these, 13 or so here, are phages that we know have a morphology which is of the Cyphiviridae. And if you remember, that's those phages that have that long, non-contractile, flexible tail. And even though the genome sequences of these are quite different from each other, or many of them are very different from each other, and I'll show you in greater detail in a minute, we can see nonetheless that in each case that the structural genes for the tail is in blue and the head is in green, the packaging in yellow here, that they form these blocks of genes that we can putatively identify in these segments of the genomes. Again, typically occupying about 25 kilobases of sequence or genome space. The order of the head and tail genes is quite well conserved. The lysis genes jump around. Sometimes they're over here towards the left, sometimes they're here to the right of the structural genes. So there's common themes of organization of the structural genes. And then there are segments which clearly seem to fluctuate. In this last case down here, BXZ1, this is not a phage with a cyphoviridal type. It's a morphotype of the Myoviridae. Remember, that's with a contractile tail. And its genome organization is quite different. And in fact, we don't know where many of these structural genes are, but they appear to be dispersed around the genome as opposed to well organized. So these general themes reflect what I showed you for ES18 and T4 earlier, and expand that to a somewhat larger set of phage genomes.